special program. First is Aldo, and then some Dutch guy who will talk about consciousness, but also more from on the one hand the neuroscience perspective, on the other from a machine perspective. Like how do we advance the theory of something complicated like consciousness by actually building it, right? But actually the real science of today is Aldo Genovesio, who's at Sapienza University. Aldo works with, he's a neurophysiologist who works with macaque monkey. And Aldo is really not a standard kind of guy, in many respects, in all positive ways, okay? Because what Aldo does, Aldo is not sticking to your standard paradigms. We discussed it also yesterday with Ranulfo. Remember, Ranulfo making that point that I have to keep the paradigm simple because otherwise I don't know what's happening. And you know what Aldo is doing is, while maintaining tight experimental control, he starts to broaden up the paradigms and really start to look at more complex behaviors in monkey and its neuronal substrate. So it's really fantastic that he's here today with us. So really looking forward. How are you going to enlighten us, Aldo?
Thank you for inviting me, Paul, and it's always a pleasure to be here after 2 years from a previous edition of a summer school. Today I'm going to talk about two aspects of decision making. One is the decision making of the individual that is making a decision about which stimulus is longer than the other in a duration discrimination task, while the second part is dealing with the individual monkey in a social context where the monkey is working with another individual that in our paradigm is not another monkey like other researchers are doing, but is a human. So we have developed, and I will talk about this in the second part of the talk, what we call a human-monkey paradigm, in which monkey and human interact in a common workspace. These are the two topics of the talk that I anticipated. Interference effects. The first part is about decision, but more specifically is about the presence of interference effects between tasks. We train monkeys, like Ranulfo was describing yesterday, in more than one task, while usually you train monkeys only in one task. Examining the neural activity of a neuron in more than one task gives us a possibility to understand more the decision process. And today I'm going to talk about two tasks recorded simultaneously. One is the duration task, and the other is a matching to sample task. The second part of the talk, I will talk about the representation of other agents. I mean the ability I'm talking about the ability of monkeys of primates to discriminate themselves from others, and not only that, but as we will see, they need also to not only to distinguish themselves from others, but also they need to make prediction about others' behavior based on their knowledge about the individual that is interacted with them. Okay, this is the first part of the talk divided in three main parts. I'm going to talk about the interference effects between tasks, as I was telling before, and I think that looking at the interference effects is interesting not only to understand how the interference is generated, but also to understand which are the neural mechanisms that underline a decision. And I will show you that looking at the interference effect allow us to understand more the decision process. And mainly what we discover is that the representation of order is very important in prefrontal cortex in order to make decisions in a comparison task, in our case, a duration discrimination task. And the second part is about the not only about the behavioral effects of the interference of one task over the other that can shed light on the decision-making process, but also because we are recording the activity at the single-cell neuron level, we can also investigate the neural representation of the relevant information. The third point is that one of the role of the prefrontal cortex is to reduce interference. And I will show you some data that indicates that there could be a neural substrate that can have a function of counteract the interference effect of other neurons that we think can produce the interference. This is the task. We have a duration task and a matching sample task. The duration task is our central task. The matching task is our control task, but in this analysis became something more than a control task. In the duration task, the monkey is required to discriminate the duration of two stimuli and of variable duration. The stimuli can have, this is very important in this talk, only two color shapes. We have a blue circle and a red square. And I will go back on this point. So the stimuli are only two. Therefore, when the monkey is watching the second stimulus, there is no ambiguity about which was the first stimulus. If the second stimulus is red, the first stimulus has to be blue. And this is, as we will say, this is very important to understand the neural activity associated to this task. We have to describe better the different steps or epochs of the task. We can start from the first epoch, that is the pre-stimulus, where a fixation point is presented to the monkey and the monkey starts to fixate it. After the monkey reaches fixation and after a pre-stimulus interval that could last 400 or 800 milliseconds, the first stimulus is presented. After we have a delay, like in the Ranulfo's task that he described yesterday. And we have the presentation of a second stimulus for a variable time and a second delay. This is important because some of the analyses that I will show you are focused on this period. After, we have a choice phase in which the monkey is required to select the stimulus that lasted longer. And the selection is made using a little table with 3 switches, and the monkey's task is to choose the switch that is below the stimulus that lasted longer. As you see here, the first stimulus lasted 1 second, the second 400 milliseconds. Therefore, the monkey's task is to select the stimulus that lasted longer, that in this case is the blue stimulus. The control task has a lot of the features of the duration task in terms of epochs, in terms of duration of events, but there is an important difference that the stimulus has the same color. So it's always either blue or red. And the monkey's task is to choose in the choice phase the stimulus that match the color and shape of a stimulus presented sequentially twice. So it's a particular type of matching to sample task. In a regular matching to sample task, you have one stimulus and you ask the monkey to choose the stimulus that is matching the first stimulus. In this case, we have a repetition of the same stimulus. So this is done with the purpose of being able to compare the matching to sample task and the duration task using a similar task structure and the similar duration of events. I will present briefly 2 example cells that describe something that is not at the center of my talk, but I want that it's clear that these cells are encoding the duration of the stimuli. And here, this is a raster plot. I think that most of you are familiar with this type of representation, but just in case you are not familiar, I will describe briefly what is a raster plot. It's, we use to represent the neural activity by dividing the neural activity recorded in different trials. So we split all the recording in different trials. And this is the first trial, the second trial, third trial, and so on. Each row is a trial. And in each row, you see small dots. Each small dot is a spike. What we do in neurophysiology in monkeys is to isolate single neurons. So we are able to isolate the activity that is associated to a single neuron from all the background. And therefore, the analysis that you will see are conducted on one neuron and on one neuron that is not contaminated by the activity of other neurons. So it's never two neurons or three neurons together, but it's one. We have, what we usually do, we align the raster plot on a certain event. In this case, the raster plot is aligned on the moment in which the first stimulus is turned off. As I showed you, in this task there is a presentation of the first stimulus, and after the presentation of the first stimulus there is a delay, and after there is the presentation of the second stimulus. Here, we sort the trials by the duration of the first stimulus. This is 200 milliseconds, for example. This is 1,200 milliseconds. So these are all trials sorted by the duration of the first stimulus. As you can see, in the delay period following the presentation of the first stimulus, you have a variation of the neural activity that depends on the duration of a previous stimulus. This neuron is represented with a higher activity, a stimulus that lasted longer. For example, you can see this is also a plot that shows you the activity as a function of the first stimulus duration. This is a second example cell that has the opposite property because the higher activity is associated to a very short stimulus of 200 milliseconds. And you see that we have very high activity after the presentation of a stimulus of 200 milliseconds. The first cell, going back to the first cell, this cell has a property that we found in many neurons, that is the presence of a climbing activity during the presentation of the stimulus. You can see the climbing activity only looking at the longer stimulus because the climbing activity starts very late during the presentation of the stimulus. We have other examples in which the activity starts to climb earlier. And we noticed an association between the presence of a climbing activity and a preference for a long duration in the memory period. So a climbing activity associated with the duration of the stimulus is often associated with the long duration with a higher activity in the delay period for a long duration of the first stimulus. We often, this is not the case, find the opposite effect with cells that show a declining activity when the cell has a higher activity associated to a short stimulus. But this cell doesn't represent this category of neurons. So I showed you that the prefrontal cortex has a representation of the duration of the first stimulus. But we don't know how this information is used yet in the comparison process. And I will go back to, for a moment, to our old results published in Neuron several years ago, where we found one important category of cell, that is category of cell that is encoding which stimulus was longer based on the color of the stimuli. This is a red longer neuron, and I will show you the property of this neuron. Here we sorted the activity in 4 categories. These are 2 rasters associated to S2 longer triad, and these are 2 rasters associated to S1 longer triad. These 2 rasters are associated to red longer triad, and these 2 to blue longer triad. And here the alignment is not anymore on the moment like before in which the first stimulus was turned off, but the alignment is on the moment in which the second stimulus is turned off. And after the second stimulus is turned off, the monkey has made the decision about which stimulus was longer, and we see a neural representation of the decision in the period of the delay, of the second delay that precede the moment of the choice. This task is very interesting because the monkey in the second delay cannot plan any motor act because the first stimulus is presented, the second stimulus is presented, there is a delay, but in this delay, the monkey doesn't know where the red and the blue stimulus will be presented because it's randomized. So in this task, there is no association between some task features and the motor action that is required to the monkey. Therefore, in this case, the monkey doesn't know yet where the goal will be placed. Now, I show you a second type of neuron that is a little bit more mysterious, because this neuron is representing something that we don't ask the monkey to represent in an explicit way. The monkey's task is just to tell us if a red stimulus is longer or shorter, because we will present a red and a blue stimulus. So this is a different task from the Ranulfo's task. In the Ranulfo's task, the monkey has to say if the first stimulus has a frequency of stimulation higher than the second. But here, the order is not a part of a requirement for the monkey. The monkey has just to decide if a blue or the red stimulus is longer. But we find this representation of relative duration based on order. So we call representation of relative duration based on color, the representation that I showed you before. We call this representation based on the order of the stimuli. And to examine briefly this neuron, you see that these neurons have the greater activity here and there, corresponding to all the triads in which the first stimulus is longer, independently of the color. We have cells that have an interaction between relative duration based on color and order. This is a more pure cell that is representing the first stimulus as longer than the second. And when we found this representation, we formulated the hypothesis that this can be an intermediate representation to a later decision. So our idea was that we first have a comparison based on order. The first stimulus is longer or shorter than the second, and after this comparison leads to another representation that is which stimulus, red or blue, is longer. But the presence of neurons of this type is not enough for us to think that these neurons are involved in the comparison. In the prefrontal cortex, it's not enough to find a neuron that is representing a certain type of information to conclude that this operation is performed by the monkey in a relevant way. This could be simply, this cell could represent simply an activity that has no function and is simply irrelevant to a task. And I will show you one example about the possibility of prefrontal cortex neurons in these tasks to represent irrelevant information, information that is not relevant to the task. It can be relevant for example, for a monitoring purpose, but is not really required by the task to be used. And so our next slide will show you an analysis that looked at the representation of irrelevant information with the goal to show you that we really don't know when we find a neural representation that this representation is really used in the computation that we are studying. And this is a different task. I'm not going to talk too much about this task, but this is a distance task where the monkey has to apply a different rule. You see that now the stimuli are at a different distance from the center, and they have the same duration in this task. And the monkey's task now is to choose the stimulus that was farther from center. You see that the red is farther from center than the blue. The two stimuli can be both on top, or either on top or on the bottom. And the structure of the task is very similar. We have a first delay, second delay, but we are not interested now, today, in the results from the distant task. I want just to show you that in this first epoch of the task, the monkey is representing information about the previous features of the previous trial's features, that the monkey is never required to maintain in memory. And these are, very briefly, the results. We looked at several variables, and we found that 28% of neurons are, for example, maintaining in memory the previous outcome. So these neurons are representing in memory if the previous trial was successful or not. Knowing that the previous trial was successful, it has no use for a monkey, because after an error, the next trial is not a correction trial. So it's not the same trial where we ask the monkey to correct his mistake, like in other paradigms we do. In this task, there is no correction trial, so each trial is independent from the others, independently from the fact that the monkey made a mistake or not. So there is a complete independence. So the representation of the previous outcome is irrelevant to the task performance, as it is irrelevant the representation of the previous goal location and object. So we have neurons that are encoding where was the goal, to the right or to the right goal, to the right or to the left, and where it was the previous object, where the blue was located, to the right or to the left. We don't find any representation of other parameters like the distance of a second stimulus from the center, or the position of the second stimulus, or the color of the second stimulus. So the only information that is encoded, irrelevant information is encoded, is about the previous goal and outcome information. So this is, in this context, just to show you that to find the representation of relative duration based on order is not really convincing that this information is really represented with the purpose of comparing the two stimuli. And we can compare two models of decision making. And the only way for us to disentangle one model from the other, I will show you, is to look at the matching to sample task. So the relative duration task per se is not able to tell us much about about something in support of the first or in support of the second model. So we have one model that we call order independent, that doesn't require order information, and we have another model that is an order dependent information that requires order. The two models are not incompatible. My point is to see if in addition to an order-independent model, we have some support about the possibility of having also order-dependent computation. This is the first model, is an order-independent model or scheme. This is a very simple scheme about a possible comparison process. This model is assuming that the duration of a red stimulus and the duration of a blue stimulus are compared by a comparator, and the output of this comparator is just the stimulus that was longer. It takes as input the red duration, the blue duration, and the output is just the stimulus that was longer, in this case the blue. This is the same model, but applied to the case in which the red is longer. Red duration is long, blue duration is short. The monkey's choice is the red longer task. This is a very simple model, and this is what you should expect, knowing the features of the task. But there is a second model that can explain in a more complicated way the decision-making process. In this model, we have an initial phase of comparison that is based on order, where the two stimuli are compared based on order to judge if the second stimulus duration is longer or shorter than the S1 duration. Let's assume that S2 duration is longer than the S1 duration. So we are in S2 longer trial. After we will examine the other case in which S2 is shorter. Okay, now after this computation, we have this output, S2 longer. We know that the second stimulus is longer. What do we imagine to do with, we can do with this information? We think that what happened later is that after you establish that the second stimulus is longer, you need to look at the S2 color. And after you have these two information, S2 is longer, S2 is blue, I choose blue. So if S2 is longer, I choose the color of S2. When S2 is shorter, and let's assume is blue, now we think the monkey, I will show you why we think that, could apply this rule. If S2 is shorter, choose the alternative color as goal. Not choose simply the color of the first stimulus, but the alternative goal, because we think that the monkey doesn't need to maintain in memory the duration of the color of the first stimulus. And this will be more clear when I will show you first the behavioral result and after the neural result that support this model in contrast with the other. So to summarize this part, we think that there is an initial comparison based on order, and after the application of a rule. If the second stimulus is longer, choose its color as goal. If S2 is shorter, choose the alternative color. That means you see the second stimulus, this is shorter, you don't even need to remember which was the color of the first stimulus, because if the second stimulus is blue, you need to choose red, because blue and red are the only colors available. And we can make a prediction based on this model. So we will consider now potential errors predicted by this model in the matching to sample task, assuming that, obviously, I will show you the percentage of errors that are very low in the matching to sample task that indicate that, obviously, in the matching to sample task, the monkey is applying the matching to sample rule. But we think that in addition to apply the matching to sample rule, the monkey is under the effect of the interference from the duration task. And the interference will appear based on the order model or order-dependent model that I showed you before. And this is our prediction based on the order-dependent model. This model predicts that the choice of a correct goal when the second stimulus is longer. So when the second stimulus is longer, so we are in the matching to sample task. The second stimulus is longer. We can let's assume that the monkey is applying the duration task rule, and the second stimulus is blue. The monkey will choose the color of the second stimulus. Good, because this is the same response that the monkey should give in the matching to sample task. Choosing the color of the second stimulus is always correct, because the color of the second stimulus is like the color of the first stimulus. So the application of a task duration rule is favoring the correct response. So the monkey will have, we think, more chances to be correct for two reasons. One, because it's applying the matching to sample rule, and plus because it's applying the duration task rule that, when the second stimulus is longer, is also favoring the same choice of the duration task rule. But the problem appears when the second stimulus is shorter. When the second stimulus is shorter, let's assume you have S2 shorter, red. You have, for example, you are in a matching to sample task where you have a red stimulus of 200 milliseconds and a red, the first stimulus of 1000 milliseconds and the second stimulus of 200 milliseconds. If you apply the duration task rule, you would choose the color of the alternative color to the second color. The second color is red. The alternative color is blue. So the monkey would choose the alternative color as goal. So you would have two conflicting rules. One is choose the red, because the second stimulus is red. This is a matching to sample task. Red, red, choose the red. The other is the task duration rule. If we are correct, it is favoring the wrong response, because the application of the task duration rule, as we hypothesized with the model that is order dependent, is choose the alternative color to the second stimulus when the second stimulus is shorter. And I will show you the behavioral evidence that we have supporting the order-based model. And this is our behavioral, okay, first let's look at the duration task. We divide the trials in S2 longer trial, S2 shorter trial in the two monkeys, and we look at the percent of error. Do we see a difference? No. So in the duration task, no trial is more difficult than the other. Doesn't matter if the second stimulus is shorter or longer, the percentage of error is comparable. Now, in the match to sample, if there was no interference, you should see the same. You should see the percentage of error that is the same, independent from which stimulus was longer, because the percentage of error doesn't depend on which stimulus is longer. But here it came the surprise, because we saw that in both monkeys we had this higher number of error in the S2 shorter trials. And I will go back of one slide to show you again the model. This is the case in which S2 is shorter, and we assume that the monkey is choosing the alternative color. So the order-dependent model is supported by the presence of this type of interference effect that cannot be explained by an order-independent model. So in the matching to sample task, there is a double number of errors in S2 shorter trials than S1 longer trial. There is no difference in the duration task. I'm going to show you now two example cells. One cell is the cell that the type of cell that you would expect to be present if there was no interference. And we have these cells. It's not that all the cells show a representation of a relative duration based on order in the matching to sample task. In fact, many cells, I will show you the proportion, are representing the relative duration only in the duration task. And here we have a matching to sample task on the top, on the bottom we have the duration task. We align the activity on the second stimulus off, so you are seeing the delay period, the second delay period in gray here. This is the period that we compare, and you see that we have higher activity in this period than in this other period. So, as the cell that I showed you before, this cell is encoding with a higher activity the S1 longer trial. But if we look at the matching to sample task, we don't see any effect. This is a cell that we would expect to find only these cells if there wasn't any interference effect. After we found this behavioral effect, now we want to have a confirmation at the neural level that there is in prefrontal cortex, because it could be in another area, but we are studying only prefrontal cortex. We want to see if in prefrontal cortex there is a representation of the information that we suppose could be causing the interference. And here I show you a second type of neuron. This is, I wanted to show you this neuron because this is the very surprising neuron. Because this neuron is representing the relative duration only in the matching to sample task. This is very, very difficult to understand because you would imagine neurons that are encoding S1 longer or shorter in the duration task and with some effect also in the matching to sample task. And we have cells like that. But this is the most difficult cell to explain. A cell that doesn't represent the relative duration in the duration task. As you see, there is nothing, and it shows a very high, very high difference of activity between the two types of trials. We performed two analyses in two different periods. One is in the delay. Now I showed you rastered were aligned on the beginning of the delay. We also looked at another period that we call decision period. The decision period it starts not always when the second stimulus starts when the second stimulus starts off, but it can start like when the second stimulus is shorter, but it can start when the second stimulus is longer, as soon as the duration of the second stimulus is matching the duration of the first stimulus. So this is a decision period starts at the decision point. The decision point is the moment in which a monkey has all the available information to make a decision. So we perform the same analysis on two periods. One is the delay period, and the other is the decision period. We found small differences, as I will show you, in the percentage of neurons belonging to one or to the other category, but the main results are independent of the period considered. And now I'm looking only at the cells, let's consider the delay period, that are selective for relative duration only in the matching to sample task, only in the duration task, or in both. These are the 152 cells. I do the same in the decision cell. And after I look, how many cells are encoding only one of the two, and how many cells are encoding both. And we see that we have a majority of cells that are encoding the relative duration only in the duration task, the blue. We have a proportion of cell that is encoding the relative duration in both tasks, and we have a proportion of cell that is like the cell that I showed you before, that is encoding the relative duration only in the matching to sample task. I need to make clear that, because we will go back and I will surprise you with a different result. Because here we are looking only at the correct trials. These are correct trials. In the correct trials, we have this situation. I will show you that we can have some information about the error trials that change a little bit our idea of the results, but we cannot apply the same. These data are, these results come from an ANOVA analysis that we cannot apply on the error trials because the error trials are very few, so we will apply a different analysis. So I cannot show you the same results on the error trials based on this analysis. Here we calculate the selectivity index or modulation index. This is very, very common in neurophysiology with monkeys. We calculate the activity that is associated to the trials in which the second stimulus is longer. We calculate the activity associated to trial in which S1 is longer. We calculate the difference and we divide by their sum. We normalize. So we have a number that is between -1 and +1. Then we calculate this index for the three populations of cells. Only duration, only image to sample, and both. I will go back on this index because this index, I will use this index to understand what is going on in the error trials. I will not use any other standard analysis, but I will use this index to see if this index is increasing or decreasing for the different type of cells when the monkey is making an error. This is the index only for correct trial. And what is interesting, you said, looking at the only matching to sample task, that although some cells don't reach the significance in the duration of task, they have a similar, let's say, a similar tendency to prefer the same stimulus as longer. But you see that there are really, there is a proportion of cell, you see the index associated to these cells that are really having a very, very, very small index. So these cells are convincingly cells that really, like the cell that I showed you before, that don't have anything to do with the duration task. So it's not just that we have some variability in the data where we can have a statistical significance in one task and not in the other. There are neurons that in the duration task don't do really anything. These are, and I will show you that we can only speculate about these neurons, but we will try. Now, this is another form of representation of the data that is called the ROC analysis that tell us how much we can predict if a trial belongs to a category or to the other, looking at the neural activity. And these ROC values, they vary between 0.5 and 1. Here we have the different category of cell, duration task, and this is the duration task and matching to sample task in the decision period for the different categories of cells. It's like a selectivity index. It tells us how much the cells are really encoding one variable. And we see that, obviously, these are cells selected by ANOVA, so we expect a difference between the two tasks for the only match to sample task and the only duration task. But it's nice to quantify this difference, although it's expected. And you see that these are the only match to sample cells that have a significant difference compared to the same cell recorded in the duration task. Here you see a greater ROC for the only duration cells when recorded in the duration task compared to the same cells when recorded in the matching to sample task. And you see that the both task cells, that I didn't show you any example, but the both task cells are cells that have the same degree of coding of a relative duration. We see that there is a small increase in ROC value for the duration task, but it's not significant. This is the second delay period. We observe a very similar pattern. So it's really both looking at the decision period and the delay period, we see similar results. And now we we can speculate based on, a little bit based on these results. These results are based on few trials, unfortunately, because we don't have in the matching to sample a lot of errors, and the matching to sample task is not recorded for. Usually it's recorded, if I remember well, for 70, 80 trials. So we don't have many trials in the matching to sample task. And therefore we can only look at what happened in their errors, pulling together all the neurons and comparing the selectivity index in the error and in the correct trial. Okay, let's look at the only duration, and let's consider the selectivity index. So the selectivity index, when we look at the comparison between correct and errors, tell us that there is a significant difference between the two, with a higher selectivity index in error for the only duration cells. So the only duration cells we saw that don't have any, in the correct trial, don't represent the relative duration. But when we look at these cells in the matching to sample task, where they were not representing the relative duration in correct trial, these cells start to represent the relative duration during errors. So the cells in correct trials, the duration cells, so I repeat, the cells that are encoding the relative duration in the duration trial, when examined in the matching to sample task in correct trial, they don't show any encoding of the relative duration. But when we see an error, for effect of the interference of the duration task with the matching to sample task, these cells can start to represent the relative duration in error trial. And we see this increase. We see a similar increase in the both cells, the cells that are encoding and the relative duration in both tasks, they also increase. But interesting, if we look at the only matching to sample task, so these are cells that are encoding the relative duration only in the matching to sample task and not in the duration task, we are looking at the error in the matching to sample task and the selectivity index in the matching to sample task, we don't see a difference. These cells during error don't increase the encoding of the relative duration. Now we can speculate that while it's kind of clear that the increase of selectivity during error trials can be responsible for the interference, we can only speculate that cells that are encoding the relative duration only in the matching to sample task might have a function of counteracting the other cells and working against the other cells that are producing the interference. This is, at the moment, the better explanation that we can find about the role of cells that show the representation of relative duration only in the matching to sample task. And I think that the fact that there is no increase of selectivity during errors for the only matching to sample task supports this argument, but is not conclusive. So we can only speculate at this point that these cells can have this function of reducing the interference. And we know that the prefrontal cortex has an important function in reducing the interference. It's one of the important functions of the prefrontal cortex, the reduction of the interference, and lesions of the prefrontal cortex are causing troubles in passing from one task to the other. And so there should be cells that have a function of reducing the interference in the prefrontal cortex. These cells could play this role. So the first summary of this first part is that the interference effect indicates that an intermediate computation in the duration comparison is based on order. I downplayed a little bit the importance of this, but I think that is an important discovery together with the others, that the order information, the information about order, is very central to the way of prefrontal cortex, of a way of the prefrontal cortex to work. So order is really central to the way in which prefrontal cortex is organizing the information. And so I think that if you use a paradigm where you require the monkey to choose the first or the second stimulus, like in other paradigms, you might not investigate that because this is a task requirement. But we have a task in which this is not a task requirement, and we see that the prefrontal cortex is using this computation as an intermediate computation in order to choose the correct goal. So this is one of the results of this new analysis. And once established with stimulus were longer, it was applied the following rule. If S1 longer than S2, then choose the goal with S2 score, otherwise the one with the S1 score. So this is very interesting because we could have found different way of solving the task in the two monkeys. So you can expect that such a rule that seems very specific can be the rule found by one monkey but not by the other. But we saw this in both monkeys, and so it seems that this is really the rule that is applied when the task is created in this way, and is the more probably is the easier way to solve the task for the monkey. And yeah.
Would you expect the same rule to be happening in wild animals, not in laboratory?
I think that if there was enough experience to deal with the problem that we posed to the monkey, I think yes. But what we don't know is that, obviously here there is a long training, no? So here the monkey had the experience, because to apply this rule, you need one characteristic of a task. You need to have only two stimuli, because otherwise you cannot apply the rule. If the second stimulus is shorter, choose the alternative color. Because if you have 4 colors, so the monkey has to remember the first color. And maybe in that case, order could be less important. So it depends. So if a monkey had enough experience with a situation like this, I think that could develop also in that case. There is, I didn't use some slides. At the end, I eliminated this slide, but this is a recurrent question that people ask about neurophysiology. What would you find in a naive monkey? Would you still find this? What I can tell you is that we have a lot of representation of irrelevant information. And there is an article that I published recently in Cerebral Cortex where we looked at another period. I could just go back a little to show you the task. Here you see that you have, for example, you have a delay, one delay and one pre-stimulus. They have different duration. The pre-stimulus can be 400 or 800 milliseconds, and the delay one can be 400 or 800 milliseconds. So this duration should not be encoded by the monkeys. But we saw that more than 10% of cells were encoding the duration of this stimulus during this one period and of this delay during this period. So the temporal structure of the task is encoded, although is completely irrelevant. And I think that it's possible that even at the first experience with a task like this, some things could be already encoded, even if it's not required, because we have the representation of durations that are irrelevant to the monkey. So irrelevant information is represented in prefrontal cortex with several purposes, because obviously the monkey needs to know when things will happen, and needs to have a knowledge of the temporal structure of a task, because develop expectation about the moments in which event will happen. Okay, so now let's move to the second part of the talk.
I'll just ask you about recorded from?
Yeah.
So was there anything?
Yeah,
I noticed a lot of your cells
yeah.
in the archaeology.
Here I limited the analysis of interference effect to the cells around the principal sulcus, mostly in dorsolateral, and some were from area 8. So it's area 8 and area 46. The original studies had penetration that were more toward also premotor cortex. But here we eliminated all the premotor cells, even if they were few in numbers even before. So it's just area 46 and area 8.
And did you notice any anatomical pattern?
Here, okay, on looking at the interference effect, I didn't look because we have few cells that are recorded both in the matching to sample task and the duration task. When I have so few cells, I have to say, I don't look for area segregation because I don't feel confident in making statement. So while when we looked at with a higher number of cells, here we recorded, I don't remember, it was more than 1,000 cells in the duration task, I saw that the effects were really spread over our recording with small difference, but small differences are not too much interested to me, unless you find that something is represented in one part and something is represented much less. Could be really depending on the variability of the recording. So.
One supplemental question.
Yeah, sure.
Given that a lot of your cells were recorded from area 8 frontal eye field, and this was a very sort of oculomotor. Well, there were oculomotor components to your task. Did you
I think that we had very, very few cells from the frontal eye field. We didn't go deep enough.
Okay.
But all the analyses that I presented were with monkey fixating the central stimulus. So there is no, at least the oculomotor involvement was later in the. One thing that I didn't say is that we tried to really to split all the different mechanisms that were involved in this comparison. And I didn't talk today about the representation of a spatial goal and action. But if we move to the choice time, we have new cells that are coming to play a role, that are defining where the goal is. So we have cells that are, for example, encoding the blue on the right. And after, we have cells that are encoding only right. So we have this mixture of cells that are encoding where is the goal and where do I have to go. So this is later. And it can be an oculomotor activity there. So I'm changing completely subject, but I'm still talking about decision, but not too much about the decision of the individual monkey about its behavior, but I'm looking now at a monkey that is wondering about the decision of other individuals. So the decision-making process that I'm talking about is the decision of others. This picture gives you an idea of our setup. There is a monkey and the human. They share a touch screen. This is their common workspace. And they can interact on this touch screen. One amazing thing that you wouldn't expect if you work with monkeys is that all the monkeys that we used, they were immediately able to be involved in the interaction. There was no training I know I will indicate in which way there was no training, but something was very spontaneous in the interaction. I will tell you which part was very spontaneous. So we call this new human-monkey paradigm. Why do we use a human-monkey paradigm and not a monkey-monkey paradigm? Depends on the question. I think that we made a series of studies to show that we wanted to know whether the monkey is able to interact with humans before starting a neurophysiological studies, and to be able to interact in turns, and we proved, I will show you later, that the monkey is able to interact in turns with humans, but also we don't have neural data about that, unfortunately. Also, we show that monkeys not only can interact, but they can also And that was very controversial before our study. The monkey can learn from humans. Before our study, the current idea was that monkey could learn only from a conspecific. So I think many people thought that the best paradigm would be to have a monkey-monkey paradigm, because the human maybe was something that the monkey couldn't understand well. So we showed that, for example, if you, in one experiment, if you have two humans that are interacting with tokens and food, and one human is you have one human that is giving a token, and the other is giving food, and the same human is giving another token, and the other human is not giving food, so one token has value, the other token has no value. In this, we use this paradigm to show that when you present the two tokens to the monkey is going to choose the token that has value, because the monkey had the ability to understand the value of the token, observing the two human interaction. So so after a series of studies of this type, we found that we could use a human instead of a monkey to investigate the social cognition in monkey. There are a lot of advantages in using a human, because monkeys can be unpredictable. You can have a monkey that doesn't want to work, and obviously even a well-trained monkey one day can make more mistakes. So you have the problem of that you cannot control. Neither of the two monkeys is easy to be an independent variable of your study. With a human-monkey paradigm, the human behavior is your independent variable. You can have a human that will make a mistake. You can have a human that is always correct. So we can manipulate the behavior of a human as our independent variable very easily. And so we can, if our paradigm requires this control, the human-monkey paradigm is a good paradigm. Obviously, it's more difficult to study hierarchies and social aspect of the type. But here I'm really interested in the representation and prediction of code. So this aspect have, I think, a small role. Obviously, primates have a social life. They live in complex social field where there are alliances that also are not stable in time. They change flexibly. So they need to have a knowledge of what is called social field in which they live. And they need to understand what other group mates are doing. And their understanding of the other individuals is very important to make predictions about the other monkey's behavior. So in order to make prediction, the monkey needs to develop a social knowledge. And it's also important to distinguish the self from others. And between others, it's important that the monkey makes different predictions for different individuals based on the experience. And social knowledge is also important for altruism, is important to cooperate. You need to know which individual is going to cooperate with you. And also it's important to have a social knowledge associated to the ability to work together in some workspace. Is everybody familiar with the mirror neurons? I think that everybody here is familiar with the mirror neurons results. And obviously, mirror neurons are very important for the understanding. And, but the idea is that they are important for action understanding. You represent the same action with the same format. And using the same format may allow you to have an understanding that is not based on inference, but is very intuitive because it's built in your system about the action of others. And obviously, we could list hundreds and hundreds of studies about the importance of mirror neurons. But our question is, which neural mechanism enable us to distinguish our action from those of other individuals? So can mirror neurons explain all our need of understanding of other neurons? Or they are very important for rational understanding, but maybe we need something more. And mirror neurons, by definition, cannot distinguish one individual from the other individual because we have a representation of the action of the same form. And they don't provide the distinction between self-performed action and other performed action because the neural codes would overlap. And obviously, they don't specify which social agent is acting. And so it's very important for understanding of action, but we need also, in parallel to the study of the mirror neuron system, we need also to look for other type of social knowledge. And we think that we need to look for separate neural mechanisms. There is one very interesting study in medial prefrontal cortex by Yoshida et al. That went in a direction that's very similar to our direction. They had two monkeys working in a common workspace. One of the two monkeys in turn was the observer. You see this monkey is always in this trial with the hand on some button, and the other monkey is moving from a starting position to one of the two objects. In this task, the object that was associated to a reward was changing every few trials, and also the role of the monkey as actor and observer was changing every few trials, and they published two very interesting articles. This is one of the two that is interesting in the context of this talk, and they looked at the neurons when the monkey was observing when it was observing the partner action or when the monkey was acting. And here the task had no delay, so the monkey could choose the object associated with the reward immediately. And so they were able to study only one period, that is the reaction movement time period. And you see that this neuron was very silent, and when the other individual was making an action, this neuron was discharging, representing an action performed by the other individual. Nothing was visible, almost very small activity was visible when there was a self-action. Obviously, there was also the opposite type of cell that was involved in the individual action, but not in the partner action. What is missing here is the delay period, because you don't know if this neuron would also be involved in the prediction of the action of the other individual. And this is one important feature that will distinguish my study from this study. And as you know, prefrontal cortex is representing goals, and also has activity that is associated to monitoring functions. And so we developed a task where the monkey is required to monitor the behavior of another individual. We call this task non-match to goal task. Non-match to goal means that in each trial, the rule is to choose a goal that doesn't match the goal that you chose in the trial before. So you need to switch goal continuously. It's not an alternation because we have more than two potential objects, so we have a series of objects. So it's not an alternation, it's a switching task in which you need to choose something different from what you chose in the previous trial. This is the temporal structure of the task. We have an intertrial. We have a period in which an holding central stimulus period where the monkey is touching a central circle, and the bar appears. The bar is useful as a go signal later, as we use it to indicate for its disappearance to the monkey that the monkey can make a decision. We have a delay, and in this is the most important period of analysis, because I will show you that in this period, when the human is when it is the human turn, The monkey is just seeing the human touching the central stimulus, but can predict what the human will choose before the action. So this delay allows us to study if there is an activity that anticipates the human choices. And okay, we have a go signal, and we have several periods. We have an holding goal period where the monkey is maintaining the hand on the goal. We have a feedback period. We have different stimuli that indicate if it's correct or not. And we have a reward delivery. So all these periods have a certain duration. We introduced these periods actually of a feedback, and we had the feedback period and the holding period before the reward delivery, because we wanted also to study the errors and in terms of feedback and of reward, but unfortunately, sometimes you regret that the monkeys are too good. And the monkeys were too good, and so you have two problems in neurophysiology. You have a problem of having a monkey that doesn't learn a task, and you have a problem of having a monkey often that learns with no errors. This is also a problem because errors, as I showed you before, are very informative. So you are never happy, because if they learn, you are not happy. If they don't learn, you are not happy. So whatever they do, you are sorry that they did like that, because you wanted something different. We are never happy. So here, here I showed you two potential objects. Now to simplify the description of a task, I use this object. Let's assume that this is the previous trial where the monkey chose the yellow goal, and in the next trials I give you 6 possibilities. It can happen one of the 6. The yellow goal can be again on the same side, or could be on the other side. It can be presented together with the blue stimulus, with the magenta, or with the green. So this means that when the monkey is in this period, holding CS, the monkey doesn't know what will be the future goal. The only thing that the monkey knows is which goal the monkey doesn't need to choose. In this case, the monkey knows that the monkey cannot choose the yellow target. But here, because I will go back on this period later, because we perform some analysis on this period. Here, there can be any representation about the future goal, neither in terms of color or in terms of position. The monkey doesn't know what will be there, but is required to maintain in memory the previous goal chosen. Okay, now I show you the same basic figure indicating the two goals on the screen, but I show you in addition a sequence of trials where you have first we divide the trial in monkey trial and in human trial. This is the first distinction. In monkey, the first 2 trials is the monkey to work. In the third trial, the blue arm indicates the human, is the human to work. Then this other trial is the human to work, and this trial is again the monkey. Okay, we developed this task with the intention of testing the ability of monkey to monitor the human's action. Because if you have just a task in which the monkey is observing something, some action of a human partner, how do you know what the monkey is doing? You don't have control on what is the monkey monitoring. No? Is the monkey monitoring the action? What is the monkey doing? We don't know. Here, we have a possibility in a special kind of trial to know what the monkey is doing. The special kind of trial to do that is what we call monkey trial interactive. Obviously, each trial is interactive in the sense that the monkey is interacting with the human. So, but we use this designation to indicate a trial of a monkey after a human trial. So for us, in this context, interactive means simply monkey interactive trial is a trial after a human trial. And here we have the possibility to be sure that the monkey is monitoring the human, because because the human can make certain number of actions in sequence, and if a monkey is not monitoring the choice of a human, when it's his turn, he wouldn't know which object to choose. But you see, the monkey here, in this example, is choosing this object because this object is not, now unfortunately you cannot see this object, but is not the object that was chosen by the human before. So the monkey can perform the task after the human only, let's say, over 50%, that is the chance level, only if a monkey is monitoring the human action. So we have a behavioral control of the involvement of the monkey in monitoring the task. So the monkey cannot do something else. The monkey has to monitor the task. And I was telling you before that we were surprised about the fact that the monkey was able to interact with the human very easily. And I was talking about one aspect of interaction, that is the fact that the monkey is sitting here, the human is sitting there, and from the first trials of interaction, the human is going with the hand to make the trial. Let's say that usually in 10, 20, 30 trials, the monkey understands that when the human is moving, the monkey goes back and leave to the human the ability to make the trial. So the monkey learns to wait if a human is making an action or not. If a human is not intervening in a trial, the monkey will do the trial. This happened in really one session of training. So the ability to interact with the human in this situation was very surprising. And there is some variability in the learning of a task, of interactive task. We also saw a monkey that learned in 1 or 2 days the interactive task. Other monkeys needed much more time. So it's really, you can see really a day learning or something that goes for weeks. It really depends on the monkey. There is a big variability in the ability of monkey to understand that has to monitor the human behavior. But we saw monkeys that learned immediately. It was something unbelievable. But it depends. Okay, now this is the behavioral control that I was talking about, and we divided the trial in what we call non-interactive, that is a monkey trial after a monkey trial, and interactive, that is a monkey trial after a human trial. And we look at the percentage of correct, and you see that we have very similar percentage of error. So the monkey is involved in the task and is monitoring the human choice. So both monkeys can recall the most recent goal chosen by the human partner and apply this knowledge to make their own future decision. Okay, now I will show you some example cells. Some of these cells are similar to the cells that Yoshida found in the medial frontal cortex. And these are cells that have a different activity based on the agent that is working. You have to consider that all these cells are recorded always with the human there. So it's not that the human goes away. So the cells that I'm presenting here are recorded when the monkey is working, or with the human here, or when the human is working with the monkey. And here we studied all the epochs that I showed you before, at the beginning when I showed you the task structure. And for example, this neuron has a higher activity at the feedback time. So when we present a feedback about the correctness of the choice, only in the monkey trial, but not in the human trial. We have a difference between monkey and human trials with higher activity in trials in which the monkey is receiving the feedback. This is a human, we call human agent cell. The distinction of, in our terminology, between human and monkey indicates simply that a human agent cell has higher activity in human trial, and the monkey agent cell has higher activity in the monkey trial. It just indicates where we find a greater activity. What is important is that all these cells, either monkey or human cells, show a difference in activity depending on who is performing the task, independently of the fact that the activity is higher for monkey or human trial. Here we have the data aligned on goals onset. This is the moment in which the monkey is presented with the two goals. And so this is our delay period that I will show you with other cells later that have a difference in the delay period. And we see that we have higher activity in the human trials. This is a percentage of cells selected by ANOVA for agent, goal position, and agent per goal position. And we see that we have a representation of agent. Now, in the analysis, we have also the goal position. So these cells are also recording, as also encoding the position of the goal. And there is also in other neurons that show an interaction. And we are looking at the delay period, the reaction movement time period, the holding goal before the feedback, in which the monkey or the human is holding the goal, and the feedback period in which the feedback is given. So we have all this period, and in all this period we can find this representation. Often we see population averages in neurophysiology where you see all the cells that are involved in the representation of something, you plot them together, and you see that information is maintained for a certain time. But some of you may want to know something more, that is, are the same cells involved in representing some variable, or different cells are involved in different periods. Here we assign 3 different colors to cells that were encoding the agent, either with more activity with monkey or with human, a cell that were not significant. Each line is a cell. We sorted the cells in a way that we could follow the same cell and to see if the same cell was encoding the same variable in different period. First question. So is a cell that's encoding the agent in the delay period encoding the agent also in the feedback period, or is it a different cell? And the second question is, when a cell is encoding the agent in one epoch with higher activity, for example, for human, is encoding in another period the agent again with a higher activity for human, or these cells switch role and can represent with opposite, in an opposite way a variable. So, and for example, let's consider this group of cells. This group of cells are monkey agent neurons. If we look at the other period, these cells are maintaining the same selectivity. Some don't represent anymore the agent. If we move to the other epoch, you see that the same, only very few cells are still encoding the monkey as agent, and many cells are losing this. But new cells are coming out that are representing the, this one. These cells, all these cells are representing the agent in the reaction movement time, and they didn't show any agent representation in the periods before. So it's a format of representation that can allow you to see if how many cells are involved in different epochs, and if the cell is encoding with the same format, the same variable. And so it indicates that we have half of the cell that change from one epoch to the other, but they usually tend to maintain the same preference for the agent, moving from one epoch to the other. We looked at the goal position, and we find a similar result with also here, for example, you see these cells are, only these cells are still selective in this epoch, from this epoch, and these new cells are coming to play a role that were not here.
Aldo, sorry, could you go back one slide? I have a question.
Here becomes very difficult to understand what is going on. So we lose, visually, you can follow maybe three, four categories.
What strikes me about that, if I've read it correctly,
Yeah.
is that there's no cell where the human agent is represented from the time of CS right through to feedback.
Exactly, yeah.
Okay.
It's really, because you should start here and to see, let's see, how many of these cells, it's really, here actually I'm lost too. So the last column is more complicated. It's easier to follow for a few columns what a cell is doing. But so in fact, a good
striking thing.
Yeah, but because I think that we should use more representation of this kind of other type, because otherwise we tend to think very often, looking at the results of an article, that the cells in a population average histogram are involved from the beginning to the end. And it's really neglected, the information about, is the cell encoding, I don't know, the object stimulus at the beginning, the same cell that. So I think that this should be an information that we start to provide, maybe also for modeling purposes. It could be, maybe it's not, we don't find it very interesting, but could be interesting because if we start to show this information, maybe we understand that there are differences. Maybe there are different, maybe some variable can be represented by the same neuron for longer, others are, or the presence of a new event can trigger a switch of representation. We often don't know. So I think that this way or other ways similar to this can be useful to have a, because it's very easy to produce something like that, and people that are interested can look into it and find what they need to understand that. Okay, now, so before I sorted the trials by which agent was performing the trial, now I introduce a second category that is the goal, left or right. And so I divide. Again, we have monkey trials, human trials, but divided by right goal and left goal. So now what I'm interested in this slide is the comparison of right and left goal trials within each agent. So here we look at, here we are in the goal holding period. So we are in the period in which the monkey is maintaining the hand or the human is maintaining the hand. And we see that we have higher activity when the monkey or the human, in this case the monkey or the human here, is maintaining the hand on the right goal. And this is common to both human trials and monkey trials. So when the monkey is watching the human in the holding goal period on the right goal, it's representing the right as opposed to the left with the same format. As you see here, you can see the activity difference between between the right goal and left goal for monkey, the right goal and the left goal for human. Okay, this is the most important cell for me. This is a cell, this is, because the other cell showed the same format of representation for choosing. Because you need cell that encode what is the goal, independently of the agent that is choosing the goal, because you need to know that which goal was chosen, the position of the goal. But we have other neurons that are, these are very interesting ones, that are encoding for example, here we have, you look at the human trial. This is the delay. The human has the hand on the center. Nothing is going on. And we see that in the delay period, we have higher activity. You can see also the activity average here. When the human will choose the left goal. The left goal is not chosen yet. The human is there. So the monkey is predicting that the human will choose the left goal. But the human is not yet on the left goal. And we don't have any difference when the monkey is on the central stimulus, ready to go to the left. So before the monkey goes to the left, there is no spatial selectivity, distinguish left from right. But when the human is at the center, in the delay period, the monkey has a greater activity for associated with the future choice of the left goal. So this looks like a predicting activity because anticipates the human choice. This is a different type of cells. Is a cell that, because in this task, ALPHO is not required. The monkey is not required to remember the previous position. But as I showed you before in the other task, you remember I showed you cells that were encoding irrelevant information, previous outcome, previous goal position, previous goal object. Here we see cells that are encoding the previous. Here we are at the beginning of the trial. We are sorting the trials not by the future goal, right or left, but by the goal position that was selected in the trial before. And we see that we have a higher activity when the previous goal was left than right, but not in the human trial. So we have also web neurons that encode the previous position specifically for one agent or the other. Here we did the postdoc analysis to identify cells that are selected for goal positioning only in monkey trials. They are the majority with the exception of the oldie goal period where they are not. Goal position only in human trials. This, light orange, and goal position in both, yellow. So we have a percentage of cells that are encoding the goal position only in human. And the most interesting for me are these cells that are showing, at least considering the design of the task, the most interesting, because I think that every activity has its interest, but the activity that was interesting me the most was the activity in the delay of this only human cell that are encoding the future human choice. Yes?
Are those cells like encoding also errors? Like what happens if a human makes a mistake?
This is the problem that we would like to look at errors, but we have very few errors in monkeys, and the recording didn't last too long. So we couldn't have also a condition in which the human made errors. Because I think that this is very critical in a task like this. If you start to have a task in which the human is making errors, you reduce the possibility of monkey to predict the human behavior. So you need to do in a very controlled way. So at least in different periods of time, different days, different weeks. The best strategy would have been to study the interaction in a condition of 100% of ability to predict, and later introduce, but we didn't have the time to introduce in this experiment.
Some prediction error type of activity?
We are running other experiments where we control the error. So where we want to to have different type of individuals, let's say, some that make always error. And we want to see if we are going to dissociate what the monkey would do from what the monkey predicts that the human would do, dissociating. I mean, the fact that we have neurons that are encoding the human choice, but not the monkey's choice, speaks in terms of prediction, because this prediction of what another individual is doing, but the next step would be to have errors, but in a controlled way, where we need to maintain possibility of a monkey to know what the human is. Otherwise, there is no prediction. If you lose that, and so we should be very careful about introducing humans making errors. As a summary, these results show that dorsolateral, we have also some neurons in ventrolateral prefrontal cortex. Let's say that the lateral prefrontal cortex has neurons with different properties important for social interaction. We have neurons that are agent-specific. We don't know yet if these cells would be, are encoding that somebody else is acting independently on the identity of the person, or if these neurons are encoding this human is acting. And if this agent-related activity is specific to individual humans, or if it just means somebody else and is acting, it's not me. We don't know. So we have neurons that encode the chosen goal for both agents, and these neurons are more ambiguous because they are representing the same goal. It's possible that these neurons are encoding the goal of the monkey when it's monkey's turn, and are performing like a mental simulation of a humans. But it's very difficult to think, to be sure that this neuron, but it's also possible this neuron is making a prediction in one case and be involved in the choice in the other. So we have some ambiguity in understanding the neurons that are representing the goal for both agents. Neurons that encode the human goal indicate that monkeys can predict the goal that the human agent will choose. The prediction of the other's choice would suggest a form of understanding on the intention of other agents. So we have the idea of continuing to record several frontal cortex area, to adding some more variation of the task, like the one that I indicated, to understand, but with a special focus on prediction and understanding of what does it mean to represent the agent that is working with the monkey. Thank you for your attention, and if you have any questions.
Questions?
Do you see any persistent activity in any of these experiments?
Any?
Persistent activity, like neurons that sustain their firing rate after you shot
As I showed you, it's now what I showed you is not the activity. When I showed you this color, I'm just showing if the neurons are involved in several parts. There are neurons with persistent activity, and
in the first part of your talk.
In the first part of the talk. Okay, the delay is very short. It's 400 or 800 milliseconds in the other task, and there are neurons that are encoding which stimulus is farther for all the duration. Yes, in a short, because the delay that we are the persistent activity can last only 800 milliseconds. No, so it's not 3 seconds. And we see neurons that are encoding from the beginning to the end.
You see?
Yeah, yeah. But because the period is very short, we have, if we look at we have the distance task. Now I didn't talk too much about the distance task. I spoke only about the distance task in terms of encoding of irrelevant previous information. But the distance task is more, is better to answer this question, because in the distance task you present the second stimulus at a certain distance, and you have 1 second of presentation plus 800 milliseconds of delay. So you have almost 2 seconds. And when you have almost 2 seconds, you see more changing of cells involved in the process of encoding.
But did it look specific to that? Do you know what is the
No,
percentage of neurons?
I didn't quantify the. I am interested in some aspects like that in a different way now, but I didn't quantify how, because it's very, it's not normal in neurophysiology to make this quantification. No, about usually you just divide in big epochs, but maybe you look at 2 epochs and you see how many cells are encoding in both, but it's rare to have more sophisticated analysis that look at that.
You were talking at the beginning of your second part about mirror neurons.
Excuse me?
You were talking about mirror neurons
Yeah,
at the beginning of the second part. And I was wondering, what would you expect in terms of behavior in success and neural activity if you're doing the same thing with some kind of robotic arm or just monkey-computer interaction?
Mm.
So are you really encoding the
Yeah,
action itself or just one stimulus?
I don't know. Really, that is really an open question. I don't know. But maybe there is a threshold over which a robot becomes like an agent, and before the threshold, maybe if it's just a lever moving, maybe it cannot be an agent, or if it is your ICAP neurons that you are using in this, maybe the ICAP is more like an agent. So that is for sure. It's something very interesting for the future to understand if there is. If the neurons are representing other individuals based on the degree in which you can assign agency to the robot or to the human. In terms of success of the realization of the task, do you have also we had to see. Okay, I think that maybe in this task it might be the same, but in observation learning task, it might fail the monkey to learn, because, for example, one important feature of it seems, because now there are a few studies about that, but monkey comparing results from different studies, it appears the monkey can learn from somebody else when the other is eating the reward. So the learning would be for vicarious learning, would be like some kind of vicarious learning. So there could be aspects that can be important, like the fact that the other is rewarded. If you have only a lever, the lever cannot, or a bar, the bar cannot be rewarded. So maybe you should have a mouth, and the mouth. So we don't know what is how much you should humanize or make him more similar to an agent, an interactive agent, to make the monkey learning from somebody else.
Would you consider that the cells that you're detecting here are working memory cells in the sense that they would maybe have different roles in different tasks?
They for sure they may have different roles in different tasks. One feature of this cell is that prefrontal cell can have a multi. We usually present results separated, for different variables, but in fact, this is another thing that maybe we should do more often, to give an idea, to classify how many cells are involved in many computations. And a short answer is that they are more than you expect reading a paper, because we don't focus too much on that, but cells can be involved in so many computations, and it's possible. So now it seems that this is an answer, but I think that we need to look more deeply in that, because I think that certain representations are compatible for one cell. There is compatibility in representing different things, but other representations are less. And we don't really, this is a field of investigation that is not too much touched yet. I give you an example. If we look, this is not published yet, but if we look at the previous outcome, like I showed you the cell data encoding, if it's correct or not in the duration task, and if we look at these cells in the distance task, most of these cells are the same. So are involved in encoding the outcome in one task or in the other. On the contrary, if we look at the representation of irrelevant durations, like I showed you just, I didn't show you raster, but I told you that there are cells that are encoding the previous delay duration or the previous pre-stimulus duration, we show that the cells are completely independent. So it's really, we don't know when a cell can represent two things, two variables or not. So the answer is not, a little bit. So it depends really on the context, and we should learn we should try to understand why two information can be combined and the two are not. I'll give you another example. In a study that I did several years ago, was a strategy task. The monkey had to learn a strategy and choose future goal, a goal based on the strategy. And in this task, the monkey was required also to remember the previous goal, special goal. And the amazing thing was that the neurons that were encoding, okay, I need to choose right, they were not encoding, I chose right. So there was a complete independence. They were between the representation of what we are going to do and what you did. So it's really, it's really dependent on the context.
Do you have a question?
You just asked my question.
Okay. Any more questions? No, I think then we'll say thank you
The
once
last
again.
thing, also, I didn't have the slide, but probably there will be a postdoc position opening in the next month. So if people want to apply, it's not open yet, but can contact me. Thank you.
Thank you very much.